So the structure of the nucleotide consists of three different parts. First off is the deoxyribose 5-carbon sugar, and then attached on one side it will be the phosphate backbone, and then attached on the other side will be the nitrogenous base, which will be a one of four nitrogenous bases: adenine, thymine, guanine, and cytosine. And then the reason why DNA is a double helix structure is because in order to like accommodate for like the base pair matchings, so that's why it needs like two strands. And then the reason why it like twists as a double helix is because it needs to like fit in and compact really tightly. And like there are a different amount of hydrogen bondings between the nitrogenous bases matchup. Like adenine and thymine will have only two hydrogen bondings, whereas guanine and cytosine will have three hydrogen bondings in between them. And then DNA, the DNA strands of the DNA double helix, like each strand, they have like an anti-parallel nature because one side will go like this way and the other side will go this way. Like one side will have start from the 5 prime end to the 3 prime end, and the other one will start from the 3 prime end to the 5 prime end. And then this really like affects the process of replication because there will be a leading strand and a lagging strand.
So what is DNA replication? DNA replication starts with an enzyme called helicase, which unzips and unwinds and unzips the double DNA helix strand. And then like this results in two separate DNA strands. And then one, like let's say there's like a top one, that will be the leading strand, and then the other strand, the separate strand, will be the lagging strand. So then for the leading strand, first like there's this enzyme called DNA polymerase, but in order for it to actually set up the nucleotides for the leading strand, there needs to be RNA primers first. So an enzyme known as RNA primase, it comes and sets up like the first few nucleotides to start like a chain, and then afterwards DNA polymerase can follow up and then like complete the rest of the nucleotide chain. And then this will complete the replication of the leading strand. However, for the lagging strand, things are a bit different. First of all, it will go, the replication of it will go in the opposite direction of the leading strand. The replication of the leading strand goes the same way as the helicase unzips the DNA. But the lagging strand, the replication goes the opposite way because on one end, because it's the 5 prime and 3 prime ends are the opposite of the leading strand, and replication can only go one way. And then, so first what will happen, like the RNA primase, same for the leading strand, will set up like segments of nucleotide chains, like here first, and then there again, and then there again, and then like each time the DNA polymerase will come and then fill up each segment like backwards. And then like this is called Okazaki fragments, and then like after this is completed, which is like much harder than the leading strand, which is just like one go, like the Okazaki fragments will be a bit fragmented. So something called DNA ligase comes and then just like puts together the fragments. And then another kind of DNA polymerase, different from the first one, it comes and it replaces all the RNA primers DNA nucleotides because the RNA primers will have like uracil in it, and then for replication, it wants like the DNA strand, not the RNA. So yeah, that pretty much ends DNA replication.